Hi, thanks for the opportunity to present our work at this conference. My name is Peter McKinnon, and I'm here with Aditi, who will be telling you about some of the work we've been doing on Aicardi-Goutières syndrome. So I'd like to just take a moment to introduce the background to the work we've been doing. So our lab is interested in understanding how DNA damage is important as a factor in many neurologic diseases. And DNA damage happens very, very frequently to most cells in the brain on a daily basis, thousands of times a day. And these different types of lesions that occur because of different cellular processes can involve when DNA is replicated, errors that happen then, the misincorporation or the wrong placement of certain types of building blocks of DNA. Free radicals from just general cellular metabolism can cause breaks in DNA. And even fundamental enzymes that are needed for the cell to work properly can also break DNA as part of their normal function. And these thousands of errors and breaks and problems that happen every day are never seen as a problem because the cells have dedicated DNA repair pathways to correct these types of damage. And so despite the fact that this is a very frequent event, we have normal cellular machinery that's able to correct these types of lesions, and so no disease happens. Now, if there is an inherited mutation or a problem with these repair factors, then we can get some rather substantial brain diseases that happen. And these brain diseases can be developmental in origin, so neurodevelopmental diseases that happen during development and after birth, or they can be neurodegenerative events that happen after birth and progress through the life of the individual. And so what we're going to tell you about today is one example where the DNA repair pathway that stops the accumulation of these wrong DNA building blocks is mutated and is not functioning properly. And it's this defect in DNA repair that underpins Aicardi-Goutières syndrome. And so the lab has been doing some new work spearheaded by Aditi in the lab, and I'm going to hand over to her so she can tell you some of our latest findings in understanding the basis for Aicardi-Goutières syndrome. And so here's Aditi. Thanks.
Thank you, Peter. So as Peter mentioned, our lab is interested in understanding the molecular basis of AGS. So AGS is a monogenic disease which is caused by mutations in several genes that are either involved in DNA RNA metabolism or in nucleic acid sensing. One such molecule is RNase H2. Mutations in RNase H2 complex comprises more than 50% cases of AGS, and today my talk is focused on RNase H2. So what is RNase H2? RNase H2 is a complex consisting of three proteins, RNase H2A, RNase H2B, and RNase H2C. It requires all three proteins to function as a complex. So if you delete any one of protein, this complex is no longer functional. RNase H2 is required during cell division. So specifically, it works during DNA replication. And if cells, they don't have RNase H2, this results in errors during DNA replication, resulting in DNA breaks, which results in accumulation of DNA damage. So majority of AGS-linked mutations in RNase H2, they are usually loss of function mutations. So our lab is interested in understanding why do mutations in RNase H2 result in AGS. There are several labs who have worked on AGS and provided important insight into its function and why mutations in RNase H2 result in AGS. So based on this, a model has been proposed where RNase H2 loss of function result in accumulation of DNA damage. This DNA damage is then sensed by DNA sensing machinery, resulting in activation of type 1 interferon signaling. And this aberrant type 1 interferon signaling is linked with AGS. However, there are many key questions that remain unanswered. For instance, nervous system is one of the organs which is affected in AGS. But why is nervous system so susceptible upon RNase H2 loss? Another key aspect is that aberrant type 1 interferon signaling is associated with AGS. But are there other factors that are involved in AGS pathogenesis? For instance, is DNA damage responsible for certain phenotype of AGS? So my work is focused on this aspect of AGS. So we first asked, how does RNase H2 prevent disease and maintain neural homeostasis? So in order to understand the molecular basis of AGS and to design better therapies, we need model systems that can recapitulate AGS. However, there is a limitation that the available AGS mouse models, they do not recapitulate the nervous system phenotype. So in order to overcome this limitation, we decided to delete RNase H2 in the mouse nervous system. So we call this mouse model as RNase nesting tree mouse model. So in this mouse model, only RNase H2 is deleted in the brain and other part of the body has normal RNase H2. So this RNase nesting tree mouse model, it doesn't show any obvious phenotype, and these mice are fertile. However, there are occasional seizures, they have been reported in these mice. So next, we looked at the brain of these mice. So we noticed that in particular, certain region of brains were affected in these mice. For example, here I'm showing you images for cerebellum region of the brain. So this is a sagittal section of a normal mouse brain, and this region here is known as cerebellum. So we noticed that RNase nesting tree mice, we see a smaller cerebellum size compared to the control brain. When we further zoomed in the cerebellum region, we noticed that certain cell types were lost in RNase nesting tree mouse brain. For instance, here in this region, if you can see a reduced cell density compared to the control brain. Similarly, in another region of the cerebellum, this region, which is known as molecular layer, we saw loss of neural cells. So RNase H2 loss, it results in specific type of neural cell loss. And these cells are required for normal cerebellum function. So since we saw neural cell loss, we wondered if these cells accumulate DNA damage and undergo cell death. So here I'm showing you images for histological images for DNA damage. So red represents a marker for DNA damage and green represents cells that are actively dividing. So we noticed that upon RNase H2 deletion in the brain, in the cerebellum, we see accumulation of DNA damage. And this DNA damage was mainly in the cells that are actively dividing. So this is consistent with RNase H2 role during DNA replication. So we also assayed for cell death, and here the green signal represents dying cells. So we see enhanced accumulation of dead cells in RNase nesting tree brain compared to the control brain. So this suggests that upon RNase H2 deficiency, cells accumulate DNA damage and then they undergo cell death. In AGS, inflammation is a key feature. So we also wanted to understand if these mice have inflammation in the brain. To test that, we performed gene expression analysis. So here I'm showing you data from control versus RNase knockout cerebellar tissue. So the blue represents genes that are downregulated in RNase nestin Cre mouse cerebellar tissue, and the red represents genes whose expression is enhanced. So we noticed that several genes that are involved in DNA, in immune signaling, these genes were upregulated in RNase nestin Cre cerebellum. We also found that type 1 interferon signaling is upregulated in RNase nestin Cre cerebellum. So here the gray bars represents the RNase nestin Cre, and these specific genes were expression was upregulated. So taken together, our data suggests that in these mice there is inflammation in the brain. So our data suggests that RNase H2 is required during early development of nervous system in mice. We don't see DNA damage in mature neurons, and the disease doesn't and the phenotype is not worsened in mice. We also found when you delete RNase H2 in the mouse brain, it results in loss of neural cells, which causes cerebral atrophy. We also noticed white matter defects and upregulation of type 1 interferon signaling in mice. So this suggests that RNase nestin Cre mouse, it recapitulates many aspects of AGS disease. So since we have a mouse model, we next asked, what is the key driver of neurological phenotype in AGS? So we wanted to understand if DNA damage is responsible, or is this the aberrant type 1 interferon signaling that is driving the neurological phenotype in AGS. So first we focused on DNA damage. As I've told you, a loss of RNase H2 results in accumulation of DNA damage, and this DNA damage is then activate a DNA repair signaling, which either repairs the damage or it results in cell death. So we wanted to investigate if we inhibit this signaling, would it worsen the phenotype? So we decided to inactivate a key component known as ATM. So ATM is a protein kinase, which is involved in DNA damage dependent signaling. So we inactivated ATM and RNase H2 together in mouse and asked if the neurological phenotype is worsened. So here I'm showing you the cerebellar images from control and RNase nestin Cre mouse brain. So then we looked at the RNase ATM double knockout mouse brain. And we see that in the cerebellum is particularly affected, and its size is significantly smaller compared to control or RNase nestin Cre mouse brain. And we also saw exaggerated cell loss in these mice. So this data suggests that ATM is activated when RNase H2 is not present in cells. So we saw this unexpected connection between RNase and ATM molecule. So when you don't have RNase and ATM together, it leads to worsened phenotypes. So next we asked if type 1 interferon signaling is responsible for AGS pathogenesis. So we decided to inhibit type 1 interferon signaling by inactivating a component of type 1 interferon signaling. So we asked if we inhibit type 1 interferon signaling and RNase H2, would it rescue the phenotype? So here I'm showing you again the cerebral images from control and RNase ATM double knockout mouse. So then we generated a triple knockout mouse where RNase ATM is inactivated, and another component which is involved in type 1 interferon signaling known as CGAS is inactivated as well. So we expected that if type 1 interferon signaling is involved, we should see rescue of the phenotype. However, when we look at the cerebellum, its size is very similar to RNase ATM double knockout, and we didn't see rescue of the cell loss as well. Although type 1 interferon signaling is restored in this mice. So Our data from this suggests that DNA damage independent of type 1 interferon signaling is driving the neurological phenotype. So, based on our work in mouse models, we think that in addition to aberrant type 1 interferon signaling, other factors such as DNA damage can drive certain aspect of AGS pathogenesis. So this work has recently been published in Neuron Journal. And with that, I would like to acknowledge this work wouldn't have been possible without the tremendous support of various people in our lab. So particularly, I would like to thank my mentor, Dr. Peter McKinnon, for his guidance and support. Various lab members, especially Susanna, who did the bioinformatic analysis, along with Patrick Schreiner, Young Dong and Young, they helped me with the histological analysis, and Helen, she generated the mouse model. I also got support from St. Jude Children's Research Hospital core facility for this work. So with that, I would like to thank you all for listening.